into it. Firstly, we're going to just go over an overview of what reference populations are. Then I'll talk about some of the benefits that our reference populations have currently provided. What is the vision for the future of reference populations, and then how interested collaborators can get involved. So our overview of reference populations. Reference populations have been around for quite some time, and simply a reference animal is an animal that's recorded for a trait, such as weight, and then we also have known relationships to our selection candidates, which can be derived from either pedigree or genomics. And then we also know a bit about those animals, so we have known management information. So you can see here in this diagram, we've got a group of animals that form our reference that have been measured for a trait and also genotyped. And then that allows us to take a DNA test of our industry animals and then reference those animals back to the information that's been recorded in our reference population by matching the patterns that are across the DNA. So why are we interested in using DNA technologies? DNA technologies are not possible without the use of reference animals. And so DNA technologies allow us to select animals at an earlier age. It allows, which basically enables faster genetic progress. We can also select for hard to measure traits or later in life traits such as eating quality, where we want to know how those animals perform, particularly for seed stock animals, without having to slaughter them. It also gives further down the supply chain better opportunity to understand the variation that exists within animals and what's genetically possible. And it also enables us to have new tools or products, particularly for commercial producers or commercial animals such as steers and heifers. If we stop selecting or collecting, phenotypes and genotypes of animals, essentially we get decay over time. So in about a 5-year period, the level of accuracy that we can predict with our DNA testing drops off if we don't maintain our reference populations. So they're integral to underpinning the use of DNA technologies in our genetic evaluations. At MLA, we have our livestock genetics program, which includes both commercial services and our R&D portfolio. So we operate the National Livestock Genetics Consortium, which is a skills-based task force that helps set the priorities for our investments and helps approve the project proposals that we invest in, and then also helps build strategies for key investment areas such as reference populations. So the two boxes above the line essentially are what sits in our R&D portfolio, and the essentials is really reference populations are considered to be an essential component alongside our work that we do on the analytical engines such as BreedPlan and Ovis. And then we also are interested in investing in new and novel R&D such as new genetic traits, new genomic tools, such and sustainability traits, etc. And then all of our R&D that we conduct through the NLGC flows through to our commercial delivery. So at the moment, we have current commercial delivery sitting within sheep genetics and BreedPlan delivered through ABRI. And then we're also interested in developing new commercial opportunities such as new DNA tools for herd profile or commercial tools, multi-breed tools, and new delivery models for genetic products, tools and services. So this, the NLGC has been operating since 2016, and we've run 5 project calls since then. And this is a breakdown of the type of investments that we've actually pursued through the NLGC. Now, the biggest box here is our reference population. So it takes up a large proportion of MLA's genetics portfolio. And then also to add, we've got this green box here, which is trait measurement. So it includes things like collection of methane or immune competence. And those projects are actually enabled by the reference populations that we run. So basically over 2/3 of the portfolio are connected to our reference populations that we've invested in over time. So what do those reference populations look like? So we've had a few flagship projects. So I'll just work through these. Repronomics is our northern beef or northern industry project. It's been running and operating for around 13 years. It's recorded around 12,000 calves, over 400 industry bulls have been represented and recorded roughly 20 to 30 traits across those animals, and it represents Brahman, Santa Gertrudis and Droughtmaster. The newest iteration of that also includes Brangus and Bradford in there as well. We've also had our Southern Multi-Breed project with New South Wales DPI that's been running for the last 5 years. In that project, we've aimed to collect 8,000 calves. We've had multiple industry sires included across 6 different breeds. And then for our sheep reference population, so we've had the Sheep Information Nucleus that was originally run by the Sheep CRC, and then MLA took over that in 2012 as the Sheep Reference Flock. So it's now been running for between those two projects around 18 years. We've recorded 40,000 lambs across those projects and represented around 2,000 industry sires. On top of that, we've also had some collaborative investments, particularly with breed associations for their beef information nucleus or BIN projects. So we've had collaborations in this space with the steers that have come out of Repronomics. We've also worked with Angus, Limousin, Hereford over a number of years as well to collect breed-specific data for those different beef breeds. The other thing that these populations, which we'll talk about a fair bit throughout the webinar, is the extra trait measurements that have been able to be collected over these projects. So we'll go into more detail on those as we go through. But the other thing that's also worth mentioning is outside of these core projects, our industry has also contributed to our reference population animals, or the size of our reference, by recording animals within seed stock herds and flocks for multiple traits and then taking a genotype on those animals. And that's of benefit to the producers that are doing that, particularly because the more related you are to the reference, the higher accuracy that you will obtain from using DNA technologies. So it's not just these core projects, although they are quite integral to underpinning our national genetic evaluations, but there's also been a large amount of contribution from industry personnel as well. Okay, so that leads on to what are the benefits of reference populations. So MLA have recently completed an impact assessment of our genetics investments through the NLGC, and basically the number that we landed on was 348 million. So that was generated by understanding the level of adoption in both beef and sheep, understanding the level of genetic progress that it's been made through both beef and sheep based on our industry indexes, and then also modelling that across our national herd and flock numbers. And so since 2016, we modelled that over a 25-year time horizon because genetics obviously is permanent and cumulative, which means it has an impact over a number of generations. And we mapped that out for 25 years, and we're predicted to have an impact of 348 million. Now, as you saw in that pie graph, over 2/3 of that benefit is linked to our reference populations in the implementation of DNA technologies, which is really good to see that flow through to industry is actually having real benefit. Now, if we translate that into what have our reference populations achieved, DNA technology is now included in all of our major beef and sheep analyses through the use of single-step genetic evaluations, which basically means that we're including DNA technology in the calculation of our breeding values alongside all of our other information like phenotypes and pedigree. We're also better accounting for the heritage of those animals, so rather than just assuming that 50% of the genetics comes from the mother and 50% comes from the father, we're able to better account for the genetics that are passed down from the grandparents and great-grandparents. We also have been able to collect hard to measure traits such as eating quality and fertility. So particularly for sheep, we've been able to select and measure eating quality traits in the reference population without seed stock animals having to be slaughtered. So that's provided, and you'll see an example of that next, of the benefit that's provided. And then also fertility is another one where they're quite later in life traits, and so the collection of that information has enabled the selection of younger bulls that have more accurate fertility information on them. And this has also led to or translated to real on-farm productivity benefits. So we've been able to improve our lambing percentages and number of calves weaned. We've also been able to improve nationally the kilograms of lamb and beef turned off per hectare per 100 mils of rainfall. And ultimately, we've also been able to do that whilst improving the quality of the product as well. So if we look at the sheep case study or what we've done in the sheep space in terms of eating quality. So in 2007, we introduced, or that's when the CRC project started and ran until 2012. And by the end of that project, we were able to generate blended breeding values where we were able to build a breeding value based off pedigree and then build a breeding value based off the genotype and bring those two together. In 2013, MLA took over the reference population as the MLA reference flock, and we started running an additional project. Now, in terms of eating quality on this graph here, we've got in the yellow is intramuscular fat, in the green is shear force, and in the blue is lean meat yield. So in 2016, part of the second project, we were able to implement the first single-step carcass traits for terminal breeds. And you can see here that that aligns quite nicely with when we started to be able to turn those trends around for those particular traits and drive them in the right direction. So shear force was going up, which was not favorable, and intramuscular fat was going down. By providing those tools to our breeders, we were able to start to select for those traits and turn those trends around. We then started the second iteration of the reference population in 2019, and as part of that project, we included eating quality traits, or as part of that work, we included eating quality traits in all of our industry indexes, and we really started to rapidly see those trends being turned around and essentially finding those curve benders that were able to perform for eating quality and perform for lean meat yield at the same time. And then excitingly alongside that, where that red dot is actually the first grid that was released, commercial grid that was released by GMP that actually accounted for eating quality traits in their pricing grids. So back in 2007, we identified that these traits were heading in the wrong direction and we needed to enable the capacity of the use of DNA technologies. And over that time, it has led to a completely changing direction of those genetic trends, which then aligned with the commercial reality of being able to implement a grid. So I think that's a really good story of how it has all come together, and it has taken a number of collaborators and industry partners to get there. So it started off with the sheep CRC, and then there's been a number of collaborators, particularly UNE and Murdoch Meat Science, the Department of Primary Industries, AWI, and also our sheep genetics breeders along the way as well. Some of the additional benefits of reference populations are really worth noting. So there was a large investment called ArmTech, which essentially set out to find the technologies that we could measure some of those key traits in an abattoir or at processing speed. And the beauty about our reference population is we were able to use that group of animals to test and validate those technologies. So So far, some of the released technologies like DEXA, SOMA, and MEQ have all been trialled and tested using resource flock carcasses and animals. And more recently, we've been using those animals to test live animal technologies for measuring IMF and muscle and fat traits on the live animals as well. It's also assisted with the development of new traits. So throughout the process, we've particularly recorded things like immune competence and methane. And the beauty about that is it allows us to correlate those new traits to our production traits that we're already recording and interested in, and also understand the variation in those traits and the ability we have to select for them, plus then build the reference that we need to develop commercial DNA tools. Other traits that we've been able to collect include some of our meat science traits. So we've worked with our MSA team on using these animals for sensory. So not only has that helped build the MSA models that are used to underpin a MSA model for sheep meat, but at the same time, it has also allowed us to use that information to determine whether or not we can have a sensory trait available to select for, which allows us to directly select for consumer eating quality outcomes. So that's both brought together our meat science MSA teams and our genetics teams as well. I think the other important part about reference populations is that it also helps demonstrate the variation that exists within animals across traits. So in a defined population, we can actually really see what are the genetics that are out there for certain traits, and also determine whether or not a trait is worth selecting for because there is enough variation. So that's been really important throughout the process as well. And then I think the other thing that often gets missed is the feedback loop, and I'll talk about that in a little bit more, but essentially we're able to look at those animals and then also look at our consumer insights and determine what are the next series of traits or the things that we need to look out for in the future, so that we can measure those in these populations and start to build the information we need to potentially lead to the development of new grids or new branded products like we saw in the eating quality example. I guess just to reiterate that a little bit further, this is a project map that we did, which basically demonstrates the purple projects are the core projects that we've operated. And then those animals have been used for numerous additional projects, such as recording methane, eating quality, immune competence on both beef and sheep, north and south, and have also connected to these other industry populations that have been recorded, such as the Merino Lifetime Productivity and Angus Sire Benchmarking Project. But the great part about this is that all of the data from not only these core references, but also these additional projects has flowed through into our commercial evaluations, including breed plan through ABRI and OVIS through sheep genetics. Okay, so we've seen that we've invested heavily in reference populations, and they've created a significant amount of benefit for both our beef and sheep industry. But what is the vision for reference populations moving forward, particularly now that we've enabled the use of DNA technology? So the current state of play is that we still have gaps in our reference animals, both in genotypes and phenotypes. And some traits have a high level of recording and some traits have a low level of recording. And this can be different across different breeds. Our reference populations have been quite expensive and taken up a large proportion of particularly the genetics portfolio at MLA. And we've also had some populations that are single breed and some populations that are multi-breed, and I'll talk about why that's important in a minute. I guess the other part to add is it's also a good time to look at how we're running our reference populations and what opportunities lie based on the timelines that we have for our different projects that are currently contracted. So this just basically says that by financial year 27, we have no lined up projects for southern beef or sheep in terms of our reference populations. And then particularly for our northern beef population, the latter half of that project is really focused on how we better engage with industry to help transition some of these projects from being core research projects to collecting traits out in industry as well. So we spent a bit of time with the National Livestock Genetics Consortium end of last year, working through what are some of the problems we're trying to solve with our reference populations and what have some of the challenges been. And the very similar issues came up. We've had a significant level of investment to date. MLA has invested close to $50 million in the last 10 years on reference populations. We still have data gaps, so there is market failure where some traits still have low levels of recording, particularly hard to measure traits. We also have had really good engagement from producers and research organisations, but there are also a number of partners that we haven't engaged with, and we think this is a bit of a lost opportunity, and that there is incredible potential for co-investment and collaboration in reference populations moving forward. And the other thing that really came up is that reference populations are still important to do. If we don't do them, we end up having decay in terms of accuracy across our products that we can deliver. If it's too fragmented, we can lose the data integrity, which means that we still need to have our genetic evaluations underpinned by reference populations, and also ensure that that's done in a coordinated model in the way it's developed and delivered. And so when we drill down with the NLGC, and these key themes came up that really are integral to how we move forward and build new models moving forward to address some of those challenges. So we came up with a bit of a strategy, and the vision really is to describe the genetic merit of all beef and sheep using world-leading genetic products that facilitate genetic progress and enable genetic gain. So that's everything from having highly accurate breeding values for our seed stock industry that really drive genetic progress, through to enabling the use of genetic tools for our commercial industry to help determine where they place animals within the marketplace. And so then the purpose of our reference populations and the strategy moving forward is to create a financially viable and collaborative and scientifically robust reference population model for beef and sheep that supports highly accurate breeding values for selection decisions. So the collaboration piece of this is incredibly important moving forward. To also help, I guess, provide a little bit of context, so recently, the last 2 weeks, MLA has released our new strategic plan. And within the first section of the strategic plan, it talks about MLA's bold ambitions and calls out key areas of investment that we're going to focus on. And one of those is around establishing multi-breed genetic evaluations and livestock credentials for both our beef and sheep industry. So we're really interested in driving genetic progress by investing in tools that support better breeding decisions, and that includes the use of genomic tools, which are underpinned by the use of reference populations. So we need to ensure that the models that we build moving forward also help support and underpin the use establishing multi-breed genetic evaluations. Another part of the strategy is really focused on creating and capturing value. So this is basically a step-by-step of how we can do that. So genetics plays an integral part of ensuring that the livestock that we breed are bred for a target market destination, and that those genetic credentials of those animals flow through the supply chain, through production, finishing, processing, and ultimately to our customer. And then likewise, the consumer feedback that we receive, not only on future market insights, but also on product, flows back down through to our genetics program, so we can select on those animals and continue to breed for the markets that those animals are going into. Now, to put that into, I guess, reference how the reference populations fit into this, or what that looks like in practice. So here we've got basically a picture of our repronomics animals. You can see all the mixed breeds there, so we're able to understand, collect all the information both within and across breed. And then we can collect all these additional traits, such as methane, immune competence, even nutritional value of those animals, utilize DNA technologies and feed that through the supply chain, which can ultimately lead to new branding opportunities for our partners that are at the other end of the supply chain. The other thing that allows is the ability to also validate those new technologies that we're talking about, to help underpin how that data flows back through the supply chain, and also ultimately helping improve market compliance of the product all the way through. And then additionally to that, it also provides the ability to have those genetic credentials follow those animals for life. So as an example, we could have genetic tools that for every lot that's sold on Auctions Plus, they have a descriptor of genetic merit either at a mob level or an individual animal level. So it really demonstrates that a collaborative approach adds value to everyone along the supply chain. Okay, so essentially that just gives a bit of a broad overview of where our level of investment has been, what the benefits are, and where we're heading to in the future. And so now I really just want to talk about how you can get involved as a collaborator in this new model being developed, new models being developed moving forward. So we've recently released an expressions of interest, and the purpose of that expression of interest is really to find the collaborators and participants that want to be involved in developing new reference projects and models. So really just canvassing everybody out there in industry who has an interest in this space and wants to put their hand up to participate. And that's really to help facilitate collaboration, co-investment and supply chain partnerships that we previously haven't engaged with before, really to support that long-term sustainability of reference populations, because we know how important they are moving forward into the future. And the NLGC really envisaged that the new models or projects that we develop will possibly differ from the traditional model that we've had of a core research reference population, and that's really to enable the capacity to expand and include the participation of new industry collaborators, herds and flocks. So who are we looking for? As part of a reference population, we can't do that without a number of groups of people. So we're really interested in engaging with our researchers for technical design and development of those projects, project managers to ensure that we're keeping those projects on track and collecting everything that we need to collect. The producers involved that can potentially be site hosts or are collecting data on farm that's relevant to help fill some of those data gaps. The use of research stations for, core reference data collection, engagement with processors, because both at the for beef and sheep projects, it's integral that we have the capacity to slaughter those research animals and also collect data implant on them, particularly for beef. So inclusion of working with our feedlotters so that they can go through those supply chains. Communication and extension personnel to help spread the message and the learnings that we get from the reference population process. Genotyping providers, so actually being able to work with those providers in collecting the DNA on all of those animals. And then we're also expanding and engaging with our retailers and wholesalers as well to demonstrate the value of those populations to those groups. We're also open to working with anyone who's not on this list. So things like ag tech companies who have technologies they want to validate, breed associations, veterinarians, any other group who is interested in engaging in utilizing reference populations as well. So the process from here, essentially the vision is that the expressions of interest process will attract interested parties from one or more of the categories that we just went through. The NLGC and MLA will then assist in bringing together all of those interested parties to contribute to a collaborative reference population project or projects, particularly addressing the current gaps we have in recording across both beef and sheep. So if you're interested, there's an online application form to fill out, which should only take around 10 minutes, and there's a series of questions. If you fit more than one category, you can fill out more than one category in that application form. From there, the NLGC will review those expressions of interest in August. So that application form closes on the 30th of July. And then due to the nature of the expressions of interest, the NLGC will facilitate the connection of all of those different applicants to collaborate on the development of full proposals or projects moving forward. Once we get to that process, the full proposals will be submitted to the NLGC, which they will then review and contract the successful projects, hoping to start some projects close to July 2026. So I guess just to wrap up, I've kind of pulled together this tagline a little bit, that we're aiming to ensure that we meet the consumer expectation every cut and every time. So reference populations are often seen to be a real genetic specific activity. However, it has multiple benefits for every participant along the supply chain, and we're really focused on making sure that we're meeting those consumer expectations. So just to recap, genomics is incredibly important in driving genetic progress, and that is underpinned by our reference populations. The value of genetics is sitting at $348 million benefit out until 2040. The sheep case study demonstrated that we can actually find those curve benders and build that market demand, which leads to new commercial grids and opportunities being developed. And the benefits are really for the whole supply chain gains. So reference populations really are the cornerstone to the delivery of DNA technologies to industry and our ability to ultimately improve genetic progress. So that's a brief overview and recap. I really appreciate everyone's time coming and joining to listen in, and I really look forward to seeing some of those expressions of interest flow through. So I'll just would go to some questions.
Yes, we have one question in the chat so far, Clara, from Lou Hogan. Do you want one application from each organisation or individual EOIs from people within an organisation?
Yeah, that's a really good question. So it's a little bit dependent. As an example, if there are different departments that offer really different components to the expressions of interest, so say, for example, a university that runs a feedlot and also has a research team, it would make sense for two applications to be submitted in that sense. However, if you've got just a group of individuals that will be assisting in providing multiple areas of that expressions of interest, then it will one application will be suitable. So comfortable for it to be all in the one application, unless you think there are sort of specific areas in which you can provide additional value.
That's all the questions that we have. Did anyone else want to add any into the chat now, or also feel free to please contact Clara or myself post the webinar today if there are any further questions in general, or also before the EOI closes on the 30th of July. Okay. That might be it, Clara.
All right. Thanks everyone for joining, and if there are any questions, please fire them through to us, and there'll also be a recording available to go back over if you've got any further questions, or let me know. That'd be great. Thanks everyone for your time.